First point is unawareness about the medicinal aromatic plants. अगर हम देखेंगे Kashmir is about
explain
for medicinal plants. और दूसरा जो problem है वो improper identification है. Third point, lack of gathering, processing and cultivation techniques. Fourth is marketing value and demands. Fifth is unavailability of the land. And sixth point is time-consuming process in cultivation of medicinal aromatic plants. And seventh is impact of climate change on MEPs. हमने फिर solutions देखे हैं, हम different awareness programs conduct कर सकते हैं जिससे हम different people को aware कर सकते हैं about medicinal aromatic plants. और अगर हम देखेंगे फॉर इम्प्रॉपर आइडेंटिफिकेशन, मैं खुद विलेज से बिलोंग करता हूँ. इनफैक्ट मुझे खुद पता नहीं था कि फॉर एग्जांपल मेंथा एरवेंस एंड आर्टेमिस एब्सिंथ हैं, वो मेडिसिनल एरोमेटिक प्लांट्स हैं. यहाँ पे आगे फिर मुझे पता चला उनमें भी मेडिसिनल प्रॉपर्टीज हैं. थर्ड पॉइंट इज हम डिफरेंट ट्रेनिंग प्रोग्राम्स कंडक्ट कर सकते हैं टू गिव द इन्फॉर्मेशन अबाउट द मेडिसिनल एरोमेटिक प्लांट्स. उसके बाद जो मार्केटिंग डिमांड, उसके लिए हमने देखा है प्रॉपर चैनल्स. फॉर एग्जांपल अगर कोई प्रोडक्ट डेवलप करता भी है पर उसको फिर प्रॉपर चैनल नहीं मिलते हैं उसको सेल करने के लिए, तो हम उसको मतलब प्रोवाइड कर सकते हैं वो चैनल, प्रॉपर चैनल जिससे वो अपना प्रोडक्ट बेच सकता है. उसके बाद अगर हम देखें अनअवेलेबिलिटी ऑफ लैंड, उसमें हम टिश्यू कल्चर टेक्निक या सॉइललेस कल्टीवेशन कर सकते हैं. फॉर एग्जांपल हाइड्रोपोनिक टिश्यू कल्चर, ताकि वो अपना जो प्रोडक्ट है वो एनहांस कर सकें. थर्ड पॉइंट इज टाइम कंज्यूमिंग प्रोसेस ऑफ कल्टीवेशन. वी कैन यूज डिफरेंट ग्रोथ रेगुलेटर्स, फॉर एग्जांपल IBA फॉर रूट promotion or root promotion hormones. And the seventh is impact of climate change on MEPs. We can use different greenhouses for cultivation.
तो सवाल जवाब हम बाद में करेंगे, ठीक है? इनको आप ऐसे ही रखना क्योंकि इसके बाद इसकी आधी एक्सरसाइज हम बाद में करेंगे. तो ये कॉमन पॉइंट्स लेके हम इनका सॉल्यूशन देंगे. आप सामने आ जाओ. Who is going to explain? आप या आप? Okay, you will hold the chart.
अस्सलाम वालेकुम एंड गुड आफ्टरनून ऑल. तो जो हमारे पास कुछ की इश्यूज हमने जो एक्सप्लेन किए हैं उसमें सबसे पहला है लैक ऑफ टेक्निकल नॉलेज. तो ये जो बेसिक चीज़ है, हमारे पास नॉलेज नहीं है मेडिसिनल एंड एरोमेटिक प्लांट्स की. तो उसके लिए हमें सॉल्यूशन ये है कि अवेयरनेस प्रोग्राम्स जो है वो ज़्यादा से ज़्यादा हमें कंडक्ट करने हैं. जैसे अभी यहाँ पे ट्रेनिंग प्रोग्राम हो रहा है तो हम डिफरेंट यूनिवर्सिटीज़ के साथ हम एफिलिएट कर सकते हैं. तो अवेयरनेस हमें बढ़ानी है इसके लिए. दूसरा है ओवर एक्सप्लॉयटेशन ऑफ नेचुरल स्टॉक. तो इसके लिए क्या है कि हमें कमर्शियल स्केल पे जो है कल्टिवेट करने हैं मेडिसिनल एंड एरोमेटिक प्लांट्स. इसके लिए हमें लैंड भी जो है वो सफिशिएंट होनी चाहिए. वो चीज़ें भी हमें देखनी है, जो बेसिक चीज़ें हैं मेडिसिनल एंड एरोमेटिक प्लांट्स की कल्टिवेशन के लिए, वो हमें देखनी है ताकि जो नेचुरल स्टॉक है वो ओवर एक्सप्लॉइट ना हो जाए. उसके बाद जो है जनरेशनल गैप है. तो हमने जब डेजर्टेशन में सर्वे किया था उसमें हमने देखा कि जो जनरेशन गैप है ये एक मेन इशू है, क्योंकि जब भी हम जाते हैं सर्वे के लिए तो वहाँ पे हमें ज़्यादातर एल्डर्स के बाद भेजा जाता है कि उनसे पूछा जाए. तो जो यंग जनरेशन है वो इंटरेस्टेड नहीं है उतनी, उनको जो है कॉर्पोरेट बिजनेस या बाकी सारी चीज़ें जो है वो उसमें इंटरेस्टेड है. तो जो इंटरेस्ट है यंग पीपल का वो हमें बढ़ाना है. इसमें जो एक्सपीरियंसेस है एल्डर पीपल के वो शेयर करने हैं उनको यंग जनरेशन के साथ, उनकी इंटरेक्शंस बढ़ानी है ताकि जो यंग जनरेशन भी है वो भी इसमें थोड़ा सा इंटरेस्ट दिखा सके. उसके बाद लॉस ऑफ बायोडाइवर्सिटी जो है, जो क्लाइमेट चेंज, इसमें हमें जो क्या क्रॉप डाइवर्सिफिकेशन कर सकते हैं जिसकी वजह से जो सॉइल की क्वालिटी है वो डिग्रेड ना हो जाए. या जब क्लाइमेट में हम देखते हैं इकोसिस्टम में कोई एक चीज़ पे असर पड़ता है उसकी वजह से पूरा जो इकोसिस्टम है उसकी जो क्वालिटी है डिग्रेड हो जाती है. तो क्रॉप डाइवर्सिफिकेशन की वजह से हम सॉइल भी बचा सकते हैं. इसमें हम ज़्यादातर मेडिसिनल प्लांट्स को ही प्रेफर करते हैं, तो जो नेचुरल स्टॉक है वो भी हमारे पास मेंटेन रहेगा. उसके बाद है लैक ऑफ एक्सेसिबिलिटी. जो ज़्यादातर हम मेडिसिनल और एरोमेटिक प्लांट्स देखते हैं ये ज़्यादातर माउंटेनियस एरियाज में पाए जाते हैं. तो इसके लिए भी सॉल्यूशन हमारे पास ये है कि हम इसकी कमर्शियल स्केल पे कल्टिवेशन करें ताकि हमें ज़्यादा दूर ना जाना पड़े इनको कलेक्ट करने के लिए. तो जो कल्टिवेशन है हमारे पास, ये हमारे लिए इसका सॉल्यूशन है. एंड लास्ट में हमारे पास है ट्रेडिशनल मेथड्स जो हम कल्टिवेशन के लिए यूज़ करते हैं. जो फार्मर्स है वो ज़्यादातर कन्विंस जल्दी नहीं होते हैं कि हम उनको अगर कोई नई चीज़ें एग्रो टेक्निक्स को हम उनको बोले कुछ चीज़ें जो है हमारे पास नई टेक्नोलॉजी है वो हमें उनको कुछ ऐसी वेज में समझाना है जिससे वो कन्विंस जल्दी हो सके. तो फार्मर्स को हमें नॉलेज देनी है ताकि वो जो ट्रेडिशनल है उससे थोड़ा सा हट के वो जो एग्रो टेक्निक्स है वो इम्प्लाई करें अपने मेडिसिनल और एरोमेटिक प्लांट्स की कल्टिवेशन के लिए. थैंक यू.
Good afternoon everyone, I am from Group C. The key issues for us are lack of information and awareness. Like, we don't know which are the medicinal plants and what part is used for by them. कौन सा part use होता है उसके extraction के लिए, और हम उसका जो solution है वो यही है कि skill development programs जैसे अभी हो रहे हैं तो उसमें हम सब कुछ explain करें कि ये part use होता है, इसके लिए ये machinery use होती है और इसके लिए ये market है, सब कुछ. कल्टीवेशन डिफ़िकल्टीज़ का ये है कि सॉइल टाइप देखी जाएगी और वहाँ पे इन्वायरमेंट देखा जाएगा, तो हर इन्वायरमेंट पे हम नहीं कर सकते उसको ग्रो. तो उसके लिए सॉल्यूशन वही है, स्किल डेवलपमेंट प्रोग्राम्स के थ्रू हम आइडेंटिफाई कर सकते हैं कि आपके जो लोकेलिटी है वहाँ पे आप क्या ग्रो कर सकते हो. हाई इन्वेस्टमेंट इन मशीनरीज, जो एक्सट्रैक्शन के लिए जो मशीनरीज यूज़ होती हैं वो बहुत ज़्यादा कॉस्ट करती हैं लैक्स में. तो उसके लिए जो सोल्यूशन है कि हम गवर्नमेंट से इंसेंटिव ले सकते हैं, बट उसके लिए आपको पहले अपना पूरा प्रोजेक्ट बनाना पड़ता है कि आपको दिखाना पड़ेगा कि आप इस चीज़ में कर रहे हो, तब आपको वो मिलेंगे इन्वेस्टमेंट्स. Lack of market awareness. Like, if you are cultivating everything and doing everything, you need to know where you need to sell the stuff. Like, you can do that by the government can do that by creating websites, social media platforms, and we can know the demand there. We need to know the demand to start a business, na? Availability of plant material. Like, if you are starting a business and you need lavender, you need to know where you can get the material. Like, the universities need to provide the information and the raw material because it is not easily available for a person. That the solution for that is like Kisan Mela. You get awareness and you can get raw materials from there, and it is the duty of us to give the raw material to the common man. Climate change, there is so many difficulties in the due to climate change. It is also the same point, cultivation difficulties, and for this we need to do proper research. Like, we need latest research material to. We have to do the research ourselves to give information to the next generation that you can do this here. Efficiency in extraction process. Like, most of the medicinal plants, the efficiency is very low. When you are extracting an oil, the efficiency is 0. something% only. You are collecting so much raw material, and when you are extracting, the process need to be very good to get more and more material, useful material from it. Na? So we need to, that is also, we need to research on that. Most of it depends on us only. Narrow range of distribution. The distribution of medicinal and aromatic plants is mainly depends upon the region we are living. Like, in temperate region, that Ardeen also told na, that lavender is only grown here. It is, we have the monopoly here. So we have to give them artificial environment if we want to grow that in Himachal or any cold region. We have to, there is so much advanced technology like Internet of Things that we can give them an environment to grow anywhere now. There are low yielding varieties in the medicinal and aromatic plants. By research work we can improve the genetics of them, and we can increase the yield. Thank you.
जल्दी पाएं. शो टू द ऑडियंस है सर. हाँ, नहीं थोड़ा, हाँ थोड़ा सा बाहर ये भी देखें और ये भी एक्सप्लेन करना है.
अस्सलाम वालेकुम, बिस्मिल्लाहिर्रहमानिर्रहीम. ये हम लोगों ने मिलके कुछ इश्यूज लिखे हैं यहाँ पे. तो इश्यूज ऐसे हैं कि बाकी लोगों ने सारे आपको बता दिए हैं, तो इसमें मैं आपको क्रक्स बताता हूँ. इसमें है जो इंफ्रास्ट्रक्चर है ना मेडिसिनल प्लांट को उनका भी प्रोडक्ट बनाने का, वो आता है बहुत महंगा है. ठीक है, वो महंगा है और रॉ मटेरियल कम है. रॉ मटेरियल कम है, इंफ्रास्ट्रक्चर महंगा है. ठीक है, तो जो बडिंग एंटरप्रेन्योर होता है वो कम्पीट नहीं कर पाता है जो कॉस्ट एफिशिएंट प्रोडक्ट्स हैं. तो उसमें मैं बोलता हूँ ये एक गवर्नमेंट को एक कॉमन इंफ्रास्ट्रक्चर एक क्रिएट करना चाहिए जहाँ बडिंग एंटरप्रेन्योर्स अपना जाके एक प्रोडक्ट बना के वो अपना कर सकते हैं, कम्पीट कर सकते हैं, उनका कॉस्ट एफिशिएंट बनता है. ठीक है, वो है. और मैनेजमेंट स्किल्स, क्योंकि कैलकुलेशन जो होती हैं वो इम्पोर्टेंट रोल रखती है. अगर आप कैलकुलेटेड नहीं हो तो आप लॉस में जा सकते हो पूरा. तीसरा है ये बिलीफ। अगर आपने एक प्रोडक्ट बनाया, फॉर एग्जांपल आपने सर दर्द की दवाई बनाई या कोई भी चीज़ बनाया, आप मासेस में रखते हो, उनके पास दो ऑप्शन थे, एक तो आपके वो एलोपैथी पैरासिटामोल बाकी चीज़ें, और दूसरा आपका, वो नहीं जाएंगे आपके प्रोडक्ट पे क्योंकि उनको बिलीव नहीं है। वो ऐसा नहीं है कि पैरासिटामोल की गोली उठा के खा लो और सब खत्म। आपको एक बिलीव क्रिएट करना है लोगों के अंदर, मासेस के अंदर, तब जाके आपका प्रोडक्ट चलेगा। दूसरी चीज़ है इसमें ये वैल्यू एडिशन के बगैर ये मैप्स का जो है ये लॉस में जा सकता है आपका वैल्यू एडिशन, अगर आपको स्मॉल स्केल पे करना है। अगर आपके पास प्लांटिंग एरिया बहुत ज़्यादा है तब आपका फायदेमंद रह सकता है, लेकिन अगर आपका प्लांटिंग एरिया कम है तो वैल्यू एडिशन के बगैर वो लॉस में जाएगा। दूसरी चीज़ है केज्ड एंड बाउंडेड थिंकिंग, ठीक है। जो हमारे पास अभी सारे प्रोडक्ट्स दिखाएंगे, सारी चीज़ें, ये पहले कहीं हुआ है, तो हम ये इस चीज़ को री इम्प्लीमेंट करते हैं अपने साथ। तो आप लोगों को ये करना है, एक किताबों में, इंटरनेट पे, इस चीज़ों पे, इन चीज़ों में नहीं अपना दिमाग बंद रखना है, आपको कुछ और सोचना है। अगर आपको गेम चेंज लानी है ना दुनिया में, तो आपको इन चीज़ों के अलग हट के सोचना है, तब जाके कोई चीज़ गेम चेंजर हो सकता है। बस
Actually, there's a lot more. पहले तो मैं आप लोगों का शुक्रिया अदा करना चाहूँगा for getting some wonderful ideas. ठीक है, हम आफ्टरनून में इसको कंटिन्यू करेंगे, लेकिन फिलहाल जो हमारे गेस्ट अभी आए हैं हम उनका लेक्चर सुन लेते हैं। सर तैयार। Then we will continue. Okay? तो आप थोड़ा सा ये इस साइड की थोड़ी सी ये चेयर्स खाली कर दीजिए, ठीक है। आप लोग पीछे की तरफ जाइए। हाँ, आप लोग भी थोड़ा सा Good afternoon, ladies and gentlemen. We are pleased to have with us our foreign guest, Professor Y.S. Trng, University of Jeju, South Korea. I would like to invite Dr. Khalid Masoodi for his introduction, please.
Assalamu alaikum, and thank you so much for having us here. So a brief introduction about our revered guest, Professor Yong Suk Chung. He is a distinguished associate professor at Jeju National University in the Republic of Korea, specializing in plant genomics and precision agriculture. His academic journey began with Bachelor of Science from Korea University, followed by successful completion of his Master's and PhD from University of Wisconsin-Madison, USA. And after earning his doctorate, he contributed in the field as postdoctoral associate at the Plant Science Institute, Iowa, in USA. Dr. Chung has held various academic and research positions, including the research professor at Chungnam National University and senior researcher at LG Chemical Farm Hanong Limited, where he focused on hybrid rice seed evaluation and production in Vietnam from 2017 to 2018. His commitment to research and academia is further highlighted by his role as a research professor at Chungnam National University. With a robust academic background and diverse research experience, Dr. Yong continues to make significant contribution to the field of plant genomics and precision agriculture. In addition to his academic commitments, Professor Chung has extended his global influence by taking on multiple roles. He serves as an adjunct professor at Ton Duc Thang University in Ho Chi Minh, Vietnam, actively participates in European cooperation in science and technology, and holds the position of CEO at Phytomix Corporation. Dr. Chung also leads the Environmental Hazard Assessment Department at the Tree Diagnosis Center and holds a key role as Vice President at the International Regional Development Corporation Research Center and Bioresource Computing Institute. Moreover, he acts as the Vice Director of Research for the Subtropical Horticulture Research Institute. Professor Chung's expertise is widely recognized, and he has been featured as an invited speaker and chairman at various special events. He travels a lot. He has been to Saudi, to Germany, Vietnam, and Thailand, and many other countries. So he has more than 200 publications to his name, and he recently got a grant of around 200 million INR from the National Research Foundation of Korea. And we are looking for a vibrant research collaboration with Jeju University and Dr. Chung. And with these words, we welcome you to Faculty of Forestry, sir.
Thank you so much. Before I invite Dr. Chang, I would like to request our worthy Dean, Faculty of Forestry, Professor Sajata Muskungu, for more welcome address.
Good morning to all of you. We are pleased, we're privileged to have our foreign guest from South Korea, Professor Yong Suk Chung, Professor University of Jeju, South Korea. He will be delivering as a lecture on plant genomics. Although it is a new technology to us. Sir, we are from faculty. You are in the Faculty of Forestry. Sir, we are familiar with the terms like genotype and phenotype. And this phenomics is a new science to us. Rather, we have not yet worked on it. If today you will give us the hints so that we can also work on this type of phenomics. Just for my fellow scientists and students, plant phenomics is a high throughput path-breaking area that meets all the requirements for the collection of accurate, rapid, and multifaceted phenotypic data. Now we have been collecting phenotypic data of trees, of herbs, shrubs, etc. But this is a new science how to do it. And plant phenomics is an approach to envisage complex traits that are appropriate for selection and provide relevant information as to why particular genotype can stand out in particular environmental conditions. The technique of plant phenotyping can be operated in various dimensions from the gene to the whole plant level under a specific environment and management practices. And why it is relevant to forestry, and because it identifies citrus identification, because you see climate change is happening day by day, and we are experiencing sometimes drought, sometimes more this rain, etc. It can help us to stress identification, study physiological processes, rapid and efficient screening, dissection and confirmation for understanding the genetic basis of different traits, genes and aspects. High throughput phenomics technology are essential to avoid human error and to reduce time consumption while phenotyping large germ plant populations or for confirmation of gene or trait functional analysis. So this is very important science, and it's a new science to us, and we hope that Professor will give us some insights how we can proceed ahead in this new type of science. And we welcome him to this Faculty of Forestry, and we can have some interactions also after his lecture. Let us welcome him to the dais and for giving us the lecture. Thank you, sir.
Thank you for coming. I'm Chang. I didn't expect I have this many people, so I must confess that I'm really nervous now. So I don't know how to deal with this, but I'll do my best. I forgot many things for now, but I hope I can catch up as I speak. And first I go next slide.
You can simply remove touching.
Oh, this is better. Thank you. Or something, as long as you have idea and stick to the definition, anyone can do it with small amount of fun. You see, I'm murmuring. I'm very much nervous. Okay. This is all about phenotyping. Phenomics is about phenotyping, and traditionally we used number, length, weight, color, and degree.
We all do it.
Yeah, right. I did it. And I do it even now.
We are doing it, right? We did.
And this is what we did. And another way we did, this data set generate quantitative data set. It's continuous data set, number wise. And another way was this. When we describe shape, we have so many words. And another one, scale. When we describe how much this leaf got diseased, we don't know how we can value this, 1% exactly. So easy way was scaling it. But this is good, but still we have one simple question. Let's say scale 1 is 0 to 9, and scale 2, 10 to 19. Those two are different value. But what about 9 and 10? Only one click, but still considered to be different. So this has kind of issue. It can be manageable, but it's not really accurate. And this way, if this, let's say this is tuber, if this tuber developed from here to here, it's not a problem, but this shape is formed by genetics, many genes involved. So we cannot tell I should be less close. Okay. It's difficult to say the sequence at the same time. Life is not like this form, it can be any form. So every shape, we have to name it, and it's almost impossible. So those two things have issue, and they generate discontinuous data set. So statistically, it has less power. So what we have to do, next please. We have digital measurement. This is what I do. And this is what you can do also. And could you? For digital measurement, this is what we use. Wavelength of light, the wavelength of light. Light is consists of many, how can I say, the length. And here, this small portion only, if we magnify, that's visual light. This is what we can see, and this is the light we use for measurement. But there are other wavelengths outside. And we use this and a little bit of outside as well. And for ultraspectral, hyperspectral, they use even further. But what I use is this one, is a little bit of outside. We call it multispectral. I will explain you a little later. First one, I said I use the visible light. That is red, green, blue, RGB image, we call it. Using RGB, we can create any color using these three colors. So that can be captured with any digital camera and even with this, anything. And could you? And digital camera, that image consists of pixels. They have many pixels. When we say 10 megapixel means within image 10 megapixels there. So all the image looks like this image consists of these pixels. Just a moment. Just a moment. I'm really sorry. I have to have deep breath. This is really
Would I take some water?
I'm okay. You are dean here, and so many students here. They are looking at me, and I'm like, plus, I feel a little pressure to give them really useful information. So, okay. RGB image is consists of pixel, and since they are consist of pixel, this controls resolution. This is what happens. For example, this small image, small flower, that small section, 10 million pixels, that will give you very clear image.
10 billion pixels.
Right. Since it's very small area, right? But when you see the satellite image, it covers so big land, right? So wide range. So even they have 10 million pixel, think about the size of 1 pixel. Sometimes 1 pixel is 1 kilometer, 1 kilometer. So no matter how many people within that area you wave at the satellite, all of you will be seen as one color like this. So number of pixel controls resolution. So if you want measure very tiny size, you have to think about the number of pixels. So that's what I want to emphasize from here. And using that RGB image, this is what we can do. This is just an example. The scale measurement can be converted to the continuous way. The way we do is this. We measure the whole area by counting pixels. If we have ruler next to it, 1 millimeter will be converted with number of pixels. By doing so, we know the size. By doing so, we can measure the area. And then those colors will be converted to blue. It's black. Then those will be counted, and you can see exactly what percentage is diseased. That's what we can do using this image technology. And another one, in breeding sector, we are dealing with so many numbers. Breeding is number game. In that case, we have to measure so many individuals. But when it comes to this type of the complex phenotype, there is limit to them all. And using image analysis technology captured by RGB, they can be trained using the machine learning to capture each of root. By doing so, you can count root, and you can measure the length, and you can measure the degree even. You just name it. You can do many things. And again, for breeding, you can have this type of the picture, thousands of pictures. But using this technology captured by RGB image, it can be done within 10 minutes, all of them, without error. Way less error than human eyes. So that's two things we can do, the example we can do using RGB image. And next, please. And another thing, visible light, a little bit outside, there is something else, like near infrared. We call it NIR. So each camera, NIR, red, blue, green, each camera lens captures that specific wavelength. So green, 560 nanometer, that specific spectrum. So each of them captures specific spectrum. In general, we use RGB and NIR. Those we use for multispectral driven data. And next, please. And this is how it works. The reason the plant leaf look as green is because plant does not use green. What I mean by that is when plants do photosynthesis, they observe red and blue for the transiting light energy to chemical energy. Those two wavelengths is used. But green and NIR does not use. Because of that, we can see they spit it out. They spit it out green color. That's why we can see green color. They observe those two. That's why we do not see it. If they are healthy, like this, red, blue, a little less. This more. Because those two are used for photosynthesis. So this wavelength number captured by a specific lens, and this number captured by the specific lens. Those, when we squeeze those numbers into various formula, we can make vegetative index. So based on this ratio, you can tell this leaf condition somehow. But this is the consequence is just number. This number does not tell you if this is sick or stressed or whatever. We do not know. This just shows you number is different. When you interpret it, you have to be careful. This is just ratio of those four numbers. Could you go to the next slide, please? And those are the examples of vegetative index. RGB are squeezed into these formula, and it can generate numbers. And each of them has slightly different the aspect. And next slide, please. Now, let me show you the very first paper ever in phenomics area. That was 2014. 2014, now 2023. It's like 10 years. History-wise, really short. At that time, with this technology, they published in Nature Communication, which is a really prestigious paper. At that time, this technology was fancy, but now this technology, any lab can do that if they can download a free software. Now you have free software. Next slide, please. This is what they did. They spent a lot of money to install Lennartech machine. They did, okay, in this greenhouse, each pot can be conveyed into the chamber automatically, and they take picture. But I want to emphasize this. This is automatic with a lot of money. But why not we can just transfer it? So I want to tell you, don't be overwhelmed by their size and the scale. We can move and we can take a picture easily. But anyway, they used this technology with big money and they took pictures. And their pictures are converted into pixel, and that one was used for measuring height, width, or whatever. Could you go to next slide? And this is the result. No matter what trait they were, X and Y is comparison with manual measurement and pixel measurement, image based. And they are very much highly correlated. So using that, they did the GWAS. So what they did was using image analysis technology, very simple image analysis technology, they showed the possibility to do GWAS. And since this was new concept 10 years ago, this could be in nature's the nature communication. Now this is very simple and anyone can do this. Next slide, please. And this one is the first paper in the field condition. And personally, I learned a lot from this paper. Again, this is the very first paper in the field using phenomics. Next slide, please. This is what they did. This is rice field, and this is tractor, and they put those sensors in the tractor. And instead of people go inside and measuring each of them, they just using the sensors, move, and done. And this is the result. Could you go to next slide? Above one and below one, same. The QTL result. Above one, they are the result of manually collected data. And below one, they are the result from these sensors data. And this is chromosome 1, 3, 6. Those peak locations are identical. Sensor data and manual data. And manual data, there are thousands of planets in the field. So when we go inside, only small sample. On the other hand, this whole plant, because sensor does it automatically. From there, the power of detection changes. Here, you see the peak here and peak here. Very small bumpy here, but this one has higher. Because this method can get a lot more samples. So sample size wise, this is way better. And another point is this. Hand measure, based to heading, biomass, height, grain yield, they are all correlated with each other. So if you, in this case, if you want to predict your yield size, yield amount, then you can go with biomass, or you can see date heading to predict this grain yield in this case. But here, you can see NDVI and NDRE, those two can be obtained using multispectral camera. Those are vegetative index. So one picture and four light spectrum and squeezed into formula, then you can get this number. One picture can give you this number of whole plant. And this height was measured by ultrasonic. If the sound comes faster, that means it's high, the taller plant. And those two, I don't know, but sense measure. When we see this, which one is easier? To predict grain yield, you can simply use NDVI in this case. So by doing experiment, you can figure out the association like this. Once you figure out the association, you can use very simple method to predict yield. So this is what I learned from this the paper. Now, could you go to the next slide, please? So from here, please do not laugh. This is what I have done. Let me introduce what I have done so far. Could you go to this? This one, the previous the paper, two papers, one 2014, and the other one was very similar time, 2016 or something, very similar time. After I saw those two papers, I realized, oh, it can be really easy. Then I did this. This was 2017, published. I used image analysis to estimate biomass. And this could be possible because in sorghum case, specifically sorghum case, sorghum has strong correlation between plant height and biomass. If it's taller, it is heavier. Very strong correlation. It's up to 0.99. It's like almost 1. So it's higher and taller, heavier. That's what I knew from paper. From there, I was thinking, what if I can associate with the green area and plant height? Then I can associate with the green area with the biomass. So I took picture to get this image. And next slide, please. I used this software. This is for free. You can download it from website. You can use it. Canopio. Yes. I use this the software for this paper. Could you go to the next slide? And this is what I did. I measured the height, and I took picture. And I compared those two, how much difference. And I measured 3 points. One, plant leaf like this, and some of them like this. And I measured the highest point, that's number one. Another one, I found the longest leaf from the top, and then go this way, and I measured top. And last way, there is node. This is plant, there is node. From node, leaf coming out. So I measured the highest node. And those three were measured. And next slide, please. And this is the result. You can see it's not super high, but at a certain point, you can see somehow usable. So my defense to publish this paper at the time was, this is not really accurate, not super accurate, but somehow high at the 5th week, meaning we can use this technology, very simple technology, for pre-breeding screening. When we do pre-breeding, we have hundreds of germplasm, and each germplasm consists of 20 plants in large field. Rather than taking the plant height, why not we just go and take a picture and then associate for pre-selection? Then this way can be feasible. That was my defense, and it was accepted. Even this simple idea and very simple way it was accepted. At that time, this journal was the top 12%. The reason I'm saying this is not because I'm trying to showing up, but because I'm trying to explain with that much small money, we could go that high. So what I spent on this paper for research was, could you go behind the back one, please? You see this black cloth thing? That's all I purchased. That was it. No, not. I'm just trying to tell you, anyone can do that as long as you have idea. Even it is not super fancy, but still, you can gradually make things different. That's my whole point here. And could you go next and next, please? And this one, what I did was during my PhD. One of them was soft rot resistance. I screened many germplasms of potato, and this was the result. At that time, I put ruler and cut in half and scanned, and I had it with me. At that time, I measured the width to see the degree of resistance at the time. But at that time, I felt limit because the disease symptom is not always like this. It's like this. So where I should measure? That was my, sometimes, more than sometimes it happens. So I was like, what should I do? But still, that was the only option I had. So I measured the widest area. That's how I did for this, the study. But thank God, I scanned at the time. So I met a person who can do machine learning, and he helped me capture that symptom area only. And he measured the area rather than the widest, the length. That is not consistent. So result, could you go to next one? Result was this. Look at this, please. 0.7. It's high, but it's not super high. I was really happy with this. This proves area could be better than the inconstant the length of width. So this very simple study using long time ago could be still published. So as long as, again, I will keep saying this, as long as you have idea, you can apply that to your study. And next slide, please. I put fancy title like this, sustainable agriculture. But let me explain how, why this could be possible. Could you go to the next slide? This is Jeju Island. This is located in Jeju Island, this buckwheat field, buckwheat. Here, we flew UAV with RGB camera and multispectral camera. And this is what we see, this color. But when they are converted into NDVI, it looks like this. Higher means more chlorophyll, I would say. Less, it does not really function, not the number-wise. So green looks good, and the red, not really good. Here, the big assumption is this. This field does not have any other stress other than nitrogen. That is the big assumption. Indeed, there was no disease, and since it's rain-fed, I assumed the irrigation, the water content-wise would be very similar. The only difference I assumed was nitrogen, the content in the soil. If that is the case, oh, I have to explain this also. I said the higher NDVI could be associated with chlorophyll. and I tested it. You see this X mark, that randomly collected sample, they were also, we measured chlorophyll content. And chlorophyll content and NDVI number there, higher the greener, lower the redder. They have high association. R square was 0.9. That means pretty much correlated, associated. And again, this could be possible because more chlorophyll means more observed red and blue and more speeded out the green. And it makes sense like this. If I measured something different other than chlorophyll, result could be different. But since I measured chlorophyll, it could be like this. And chlorophyll is very much related with nitrogen in soil. So if we can use this smartly, we can reduce fertilizing, nitrogen fertilizer, like this. This was my idea. If we can make grid here, A, B, C, D, E, and no 1, 2, 3, 4, 5. Now we have technology with UAV. We can send UAV and we can drop the fertilizer. If we can program to drone, only these places are fertilized, then it can be precision agriculture, targeted area. And by doing so, we can reduce the consumption of fertilizer. That's why I said the sustainable agriculture could be possible. And as you may know, nitrogen fertilizer can be observed only 5% by crop. Could be more, could be less, but what I saw in the papers, review papers, they say only 5% of fertilized the nitrogen can be observed. 95 can directly go to the water, underground water. So if we can use this very simple technology, we may be able to reduce the fertilizer and we can protect environment. And by using less fertilizer, we can emit less carbon dioxide. So that's why I could make that kind of fancy technology. But result was simple, but as long as you have idea to be connected with, then you can make a story, then you can publish like this. So simple idea and other area connected each other to make a story, and still with less money, you can do something important. And next slide, please. And this one we hired people who can do machine learning. Here in Kashmir, I strongly believe there are many computer science people. And what you have to do is just talking with them. Here is what I have. What can you do? Can you help me? Something like that. That's all you have to do. That's what I did for this study. So next slide, please. We had thousands of the pictures like this. And this is what we wanted. We fertilized silicon and non-silicon. We want to compare the number of the nodule. That's what we do. We just count number. But with this picture, how we can do much? If we do not have this picture, we cannot do 2,000 in a limited time. So we took image because of the limited time. Then our mission was counting the nodule. But with our naked eyes, with human error, how we can count them all? So we trained this image. We trained computer. By doing so, we keep increasing, and eventually we could train this accuracy. Originally, we could count the nodule only with error. But now, not only counting the nodule number, but also size and location. We have more parameters now. This simple study is one example of the importance of collaboration among different areas. So no matter how simple that the experiment could be, Still, if you combine with other area, you can make something bigger. That's what I want to say with this example. And next slide, please. And similar thing. Yes.
Can we go to the previous slide? Just
Please.
I want to know. Yes. The image which you have captured is okay. It must be one side. What
Yes.
about the other side or the four sides?
That is the drawback.
Yeah.
That's all we can have. But as you know, that we have another big assumption. If we try to put this image as much as we can show the big more area, we took picture this way. If root looks like this way in 3D, we didn't picture take this way. We took picture with this way or this way, so that we can see more area.
We can have two. We can have two pictures, front and back. That will maximize the coverage.
At the time, we didn't have that kind of opinion. We were just, this was very beginning time,
Yes.
so that's really great idea. But at the time, we were like, oh, we don't lose this data. Let's just take a picture. That was it.
But in that case, also there is a possibility of doubling the nodules.
At the time,
if it's
yes.
on the side, it will be captured both in the front as well as at the back.
If we did that, like you said, it could be really better. But
Maybe.
even like that, that's improvement. But at the time, we did not have any concept about that. We just took picture and then let's just ask help. That was it. So from there, we could get this. And this was accepted. But of course, what you said was really important aspect. And next slide, please. And this one uses the same technology using the RGB and the machine learning thing. And next slide, please. We did hundreds of germplasm, and each of them have many the individual pictures. So to sum up, many numbers of pictures. But using this way, this technology, we could get various parameters in a split second using this technology. And those parameters were squeezed into PCA like this. And you see this, the two colors, the blue and red. Red one is cultivated soybean. Blue one, wild germplasm soybean. We assumed wild germplasm would have more variance than cultivated the soybean. But result was different. From this, we realized there are huge potential to breed the root part using even with, what is this? The cultivated soybean. So that's why the title was, go to previous one please. A large root phenome database wide open the potential for underground breeding in soybean. That's how this title could be solid. So this type of the technology to obtaining parameters nowadays is universal. You can do that or you can buy software. But that type of universal technology can be connected with idea, can make things different. So again, simple method with story can do something. That's what I want to emphasize over and over again. Next one, please. And this one is ridiculously simple. I used blueberry. Each blueberry, I used star cultivar and Suji blue cultivar. 2 cultivars only. And each cultivar, 15 individuals, as far as I remember correctly. And each individual, each species 15, and each individual, high, middle, low, and each part 15 leaves, or 20 leaves, I forgot. It's written in the paper. And the replication-wise, it was sufficient. And this was the result. This is fed speed reflect the activity of photosynthesis at the given time with the given sunlight. If light is stronger, more photosynthesis activity will be happening, then higher number of speed value will be the measure. And now, height-wise, high, middle, low, and time-wise, in the morning, daytime, in the evening. And this is speed value. What I saw from this picture is this. They are blueberry, same species, but the difference was cultivar, different cultivar. Even they are the same species, if they are different cultivar each other, meaning those two cultivars from different origin. Their parents could be hot place, cold place, cold place, cold place, whatever. Different origin, background. If that's the case, different genetic background, they could behave differently. This means this can be connected with smart farm technology, because in smart farm, everything is controlled, even light. In this star, the cultivar case, in the morning, you don't need to give them high light. You just need sunlight normal way. But in this case, you have to give them more sunlight. So if we make model for smart farm using species only, just species only, that species was represented by one, the cultivar, then you can miss this type of effect. If that's the case, you cannot build the how can I say it, precise model. And if that's the case, you will consume more energy. And can you go to the next slide? Do I have that? No, I don't have it. Okay. Could you go back? I have to explain that one. Number one, we have to think about we have to study and make model based on species. This is the reason. You heard about AI. That AI use model. That model is based on big data, right? You need big data to make a model, and you need a model for AI, for the automated control. And the model, can I write something here? This is very important aspect. I really want to show here. Thank you. If your data is here, and based on this data, you make model like this. This is perfect. This is what you need to do AI in the future. So these dots represent what is this? The big data. But if your data is this way, it will give you similar line. But does it accurate? It's not accurate. If your, okay, let's this big data is combined with the two, the cultivar. One of them infect this way, one of them infect this way, and somehow combined together makes this way. If we split those two data sets, this one can grow this way, this one can grow this way. So when we deal with this cultivar within the same species, we have to develop based on this data. And when we develop AI system based on this data, you will have a lot better result compared to this. So from this data set, this result, I argued this type of thing in the paper, which could be important for smart farming in the future. So this could be accepted. And in this case, to get this kind of conclusion, all I had was speedometer and the what is this? The blueberry cultivars. And that was done one day. So basically what I spent money, the money I spent was buying the speedometer and paper and the ballpoint pen to get the data. And one day, as long as you have somehow clear the difference using the replication, and with the idea, you can do phenomics. Phenomics doesn't need to have a lot of equipment and fancy technology. Idea and story is important, as you can see here. And how much time do I have? I think I am talking too much.
Interesting.
And if anyone have the question, please let me know. And next one, please. And this one is recently done. Not very much recently, but anyway. People used image analysis for measuring seeds, many cases. But I was a little bit late for that type of research, so I was thinking how I can make this published, how I can make this the useful. And though this is the way we collected data. We have camera, and below here we the buckwheat seeds, and we took image like this. And could you go to next slide? And could you click somewhere here? I have 100 seeds in each, about 100. Oh, that's all. Oh, install video. Okay. Okay. We have 100 each slide, the picture, and we have 600 the images. So think about it. 100 seeds times 600 germplasm. That is 60,000 data points. And if we can generate 5 parameters, that means 300,000 data points. Once you get this kind of image, by simple software, very simple software, you can get that 3 on the computer power. Personal computer. 5 to 10, 15 minutes, you will get 300,000 data points. And using that, what we can do? The advantage in this case, this kind of the small seeds, there is limit to measure the seed. You cannot get many seeds. Only limit like 5 or 10 maximum. But here, 100. So number-wise, we have very much advantage. More number, more statistical power. That's our argument. And the different parameters that could not be used before, such as the area. We could measure area, but here we could measure area. More, 5 parameters. Now, could you go to the next slide? This was the paper we published with this data. In GeneBank, we go to GeneBank to get the germplasm for certain the target, the trait. We want to use that specific target trait, and that one should be incorporated the target breeding purpose. But when they provide the number that for the specific target, they just give us the mean value. But that could be dangerous, and that is my argument. Because of this, based on the result of this experiment, we found out this one, 4 different germplasm, and depending on the germplasm, the variance different. Some germplasm has very small variance, some germplasm has very wide variance. If we have only mean, what will happen if we have this? Mean is this, but what if we get seed from here? So we need to have standard deviation at least. How we can get the accurate standard deviation? We can use this type of technology. So that was my argument for this. So the implication of high variance, there are other stories in this paper, but because of this big number, we could reach this kind of conclusion. Without big data, we can say this. Yes?
What is this density?
Density was the area.
Area of the
Just
seed?
one. Yeah, area. No, correction.
Yeah.
This is area, this is the frequency. Density is frequency. The term I should explain.
Okay. Means if we under area we got number 8, so it will be the frequency of number 8, that how many times it occurs in that data set.
Exactly. So this area, this size of area has a lot higher frequency than other area size.
This is about the previous picture you showed. This is the paper
Yes.
of that.
Yes, based on the previous result.
Oh, okay.
This is the normal distribution. It can be anything. This is the normal distribution of it. We get such type of problem.
Okay, I got your point. Okay. Yeah.
So immunofluorescence, good thing, we can get more number more easily. And could you go to the next slide, please? And this one was the starting from the very kind of stupid idea. I happen to know one of my undergraduate students can deal with satellite image data. While we are talking with something, he said satellite image can be free. There are free satellite images available on the internet. That's what I was told. Then I was thinking, oh, what we can do with this, the free data. And this is the paper I did. In Jeju Island, we have certain peak of, not mountain, but kind of mountain result from the volcano activity, and very small area. But considering big area is very small area, but still to walk around, it can take several hours. So that big, that size of the area. And could you go to the next slide? We captured the image from satellite and we normalized those pictures, and using that normalized pictures, we calculate NDVI and showed in different color like this. In 2000, all of them, April 8th, April 7th, April 7th. So 1 day different each, but 1 year apart different from 19, 20, 21. So on April 19, 20, 21, and they are different. They are different. Whatever happened, they are different. And why is that so? We try to associate with many other the weather data. And could you go to the next slide, please? When we plotted with average temperature with that NDVI numbers, we had high correlation. So from this simple plot, I was thinking, oh, if this is the case in this given location, why not we can use this technology, not even technology, yeah, very simple technology for monitoring the temperature changes. If this is okay, then we can even expanding the area to wider and wider for monitoring temperature, although they do not have the weather station, since we have such high correlation. So my argument in this paper was, since they are highly correlated, even there is no weather station, we can use this for monitoring the temperature changes over time to see how much serious this global change is. So since they are solid data, I didn't change, I didn't manipulate anything. Reviewers accepted. And somehow we might be able to use this for remote area. So again, no high technology, not a lot of money, but still as long as you have idea and story, you can there is a chance for you to utilize that simple thing. And next slide, please. This is next. Oh. Yeah, this is again. I used
Tree composition.
the free satellite image, and when I said composition in forest means evergreen versus non-evergreen. Of course, within this non-evergreen, there could be grass, but at least we can see the evergreen area. Whatever region, evergreen area can be shrinked or expanded. But how we can see it in easy way, that was the thing in this paper. In summer, everything will be green, like this. And in winter, only evergreen will be in green. And if we subtract from summer to winter, then we see somehow grass and the other region. And using this way, we can monitor their changes in area easily. So although the title seems to be fancy, but the idea itself is super easy and super simple. But as long as it can be applicable, then it can be accepted. And we can use this for easy monitoring, rather than many experts are sent there.
Again, the data is from satellite.
Free satellite.
Yes.
We have been using this already, Dr. Atlat.
Guide me, please.
No.
So then,
You're going very good. I'm developing interest in your lecture. It's very good.
I try to use the simple one. And next slide, please. Yeah, the same idea. Please, next and next. And this one was done very much recently. And next one, please. To take image, we used this simple the camera inside, and that was connected with Raspberry Pi. This is very comparably cheap the material, and we use this one. And we programmed here to take picture every 10 minutes. We can control the time interval, whatever you want. And next slide. The reason I made this slide, the experiment set, was because of this. I was assigned, not assigned, I applied for a fund, and that fund was this. Can we develop a system to detect the brown hopper? You know what brown hopper is? Yeah. Brown hopper damage. Can we detect it in early stage? That was the title of the funding. And in order for us to see the possibility of that developing technology, I did this. If the plant color changes. Entire plant, every area, this color changes the same way. Say this part changing color same way, then we can do that. But if this part change color, this part changing color, and this part change later, then we cannot detect it in our stage, right? Because UAV take picture from above. If this part goes yellow first, then this part, how we can detect this vertical area, right? That was the thing I want to check. By taking picture every 10 minutes, we found this. This main part goes yellow first. And the leaf area goes late. And if we magnify that area, you see this little joint? That joint is this. So main stem, they are hanging in the main stem and soaking the sap. Result, this stem going down. And that, the effect go beyond through here. And because of this, that effect can be shown later. And because of this, we can conclude it is impossible for us to detect this disease symptom in early stage. And with this simple and the low cost test, we could prove it. And next slide, please. And the way the changing color, we can use many other, the vegetative index, we can choose what's the best, and based on that, we can make the detailed data set to test the statistics. But anyway, and next slide. And this is from above. It changes. It's not important. And next slide, please. Okay. I have several more, but how much time did I talk? About 1 hour. Okay. I think I gave you enough example, and they are about to be similar. As long as you have idea and story from the idea with small number of the data set or small budget, you can make something different. That was the whole point of me being here. Thank you very much.
Let me thank our invited speaker, Professor Yong Sung Chung from University of Jeju, South Korea. Personally, we have a meeting going on. Then I started developing, as I already told you. But it's simple. Technology, as long as it's not simple and it's conveyed in a simple manner to any of the normal undergrad student or to a common person, then that technology is not kind of useful or appealing. You presented it in a very simple way, and at every stage you told that it's very simple and everybody can do it. So I think a round of applause for that. Thank you, Dr. Khalid Masoodi, for being here today with us and providing with us an opportunity to hear from the guests. Our sincere thanks to Professor Sajjad Ahmed Gangu Saab, Dean Faculty of Forestry, for being present in this lecture. Thank you, audience. Thank you, all colleagues, for being here today and listening to our expert. Thank you very much for coming here. We look forward to more of you from your side. Thank you very much.